This is part 3 of the lecture on viruses. I got cut off when I was trying to write this last part. Some viruses have a complex shape, and one of those is the bacteriophage, the T4 bacteriophage that infects E. coli, and we call that complex shape head and tail. And it's a combination of the head, which is icosahedral, and the tail, which is helical. And here's just some different images of viruses. This is naked icosahedral, like the adenovirus, and then enveloped icosahedral, like the rabies. Here, I'll write these. This is just pictures that could be. And so the rabies is an icosahedral capsid with an oval envelope outside of that. And this one, I really don't remember what it is, but it's a helical structure with an envelope around it. And then this one is naked helical. That's it. Each one of these has got a helical shape, so it looks like a stick, but actually inside of that stick is the helical structure. All right, and then these are complex. So it's hard to tell what their shape looks like from that individual picture. But just some examples, mainly so you can see the differences for yourself. Okay, what we are going to concentrate on in this section is we're going to talk about in Bio 111 what we learned about protein synthesis in cells. Okay, we learned that DNA inside the nucleus makes a copy of itself in the form of messenger RNA. The messenger RNA leaves the nucleus and codes for the amino acids, the specific amino acids that form the protein. So when DNA produces messenger RNA, we call that transcription, and it involves an enzyme, RNA polymerase. Then when messenger RNA codes for the amino acids in the protein and the protein is produced, we call that process translation. So this is what happens in normal cells. A double-stranded DNA molecule produces a single-stranded messenger RNA molecule. And then that messenger RNA is translated into a protein, which can be 10,000 to hundreds of thousands of amino acids long. Now, in the Baltimore classification, this is the classification system we use today, there are 7 groups, and it's all based on what is the nucleic acid core, and what is the mode of transcription or the mode of messenger RNA production. So the characteristics are going to be whether it's single-stranded RNA or DNA or double-stranded RNA or DNA, and then the mode of messenger RNA production is going to be how transcription occurs in that virus. Okay, so Group 1 viruses in the Baltimore classification system contain double-stranded DNA, and as you might guess, everything happens just like we learned. Going back to this page, DNA undergoes transcription, producing messenger RNA, and then messenger RNA undergoes translation, producing a protein. So no changes needed, and the herpes simplex virus is an example. One of the herpes simplex virus is an example. Okay, that can cause fever blisters or genital herpes. Now, group 2 is single-stranded DNA. So it has one extra step added to it in the beginning. The single-stranded DNA makes a copy of itself and becomes a double-stranded DNA, makes a complementary copy of itself. So for example, if the if the DNA had this code CATG, the complementary strand would be GTAC. And so because A's, if you remember this, A's and T's are bonded together, and C's and G's are bonded together. So the parvovirus is an example, canine, which can cause parvo in dogs, which is very, very difficult to treat and often results in death. So because they have diarrhea and they get dehydrated. All right, group 3 is a double-stranded RNA. And that double-stranded RNA codes for messenger RNA, and then messenger RNA codes for protein. So it's pretty much like group 1, except the beginning molecule, the beginning nucleic acid is double-stranded RNA. And there is a virus called a rotavirus, which causes gastroenteritis. That means stomach and intestinal. Itis means inflammation. Gastro means stomach. Entero means intestines. So it just means inflammation or infection within the stomach and the intestines. Typically, this one occurs in childhood. Now, group 4 is single-stranded RNA positive. And all that means is that no transcription occurs at all. The single-stranded RNA molecule is the messenger RNA. So it's just directly converted into a protein. The common cold caused by a picornavirus is an example of a single-stranded positive RNA virus. Then single-stranded negative. The messenger RNA is transcribed from the single-stranded RNA. So this single-stranded negative RNA is transcribed to messenger RNA, and messenger RNA is translated to protein. And rabies, which is a rhabdovirus, is an example of a group 5 virus. In group 6, your single-stranded RNA virus has reverse transcriptase. So this is a retrovirus. And that reverse transcriptase uses the RNA, the single-stranded RNA, to produce DNA, and then the DNA is incorporated into the host genome, and transcription occurs there. And HIV is an example of a group 6 virus. Group 7 virus is double-stranded DNA virus with reverse transcriptase. And this one is very strange. It's kind of difficult to understand. But even though the virus has a double-stranded DNA molecule, it does not go directly into transcription. Somehow it's replicated into an RNA molecule, and the RNA can be messenger RNA, or it can be used as a blueprint or a template to make messenger RNA. And hepatitis B is an example. You know, there's different types of hepatitis. All right, so this is just to show you what happens in each group. In group 1, you have single-stranded DNA. No. I'm so sorry. I didn't put group 1 on here because group 1 is just like we learned in Bio 111. So in group 1, you have double-stranded DNA. It's converted into messenger RNA, and then messenger RNA is converted into protein. But we're really just looking at the conversion to messenger RNA, that process called transcription. So in group 2, the virus has single-stranded DNA, and that single-stranded DNA makes another strand. It becomes double-stranded, and that uses DNA polymerase, the enzyme DNA polymerase. And then RNA polymerase is used to carry out transcription, to convert the double-stranded DNA to messenger RNA. Group 3 has a double-stranded RNA, and that is used to make messenger RNA. So that double-stranded RNA is used for the transcription molecule, and the enzyme that carries that out is called RNA polymerase. In group 4, you have single-stranded RNA positive, which just serves as messenger RNA, so there is no transcription. That step is skipped. But if the molecule is single-stranded RNA negative, it undergoes transcription, producing messenger RNA using RNA polymerase, and then of course the messenger RNA is used to produce the protein. In group 6, single-stranded RNA is converted to double-stranded DNA, and that process uses reverse transcriptase, and then RNA polymerase is used to convert the double-stranded DNA to messenger RNA, and then you have your messenger RNA converted to protein. Group 7, double-stranded DNA with reverse transcriptase. Most commonly what happens is the double-stranded DNA is converted to an RNA intermediate, and then the reverse transcriptase comes in at this point and converts the RNA intermediate into single-stranded DNA, which is used for transcription to produce messenger RNA. Okay, moving on, the impact of viral infection. You should probably know, if you don't already know, you should probably know. We've learned a lot about viruses this past couple of years, almost 2 years now. Well, 1.5, but it just seems like it's been forever. But the impact of viral infection is just the symptoms that the virus gives you. So they can be very mild to very severe, like deadly, but they can't be treated with antibiotics because antibiotics treat bacterial infections. They can be prevented with vaccines, and the symptoms can be treated with certain medicines, but the virus can't be destroyed, by antibiotics. Antibiotics are useless. So it says, when the virus reproduces, the viral replication cycle. We don't call it the life cycle because the virus is not a living thing. This can cause damage to the host cell, and that's what gives you the effects of the disease. Cytopathic effects refers to the effects on each of your cells. So it can change your cell functions or even destroy the cell. The symptoms of viral diseases result from the immune response to the virus. And the immune response typically includes things like fever and, maybe cold symptoms and that kind of thing. Pain, swelling. Okay, so the replication cycle, some infected cells, like the ones infected by the common cold virus. Common cold virus is the rhinovirus most of the time, even though we learned that the adenovirus can cause the common cold, the picornavirus can cause the common cold, but the main one, the primary virus, is the rhinovirus. And what it does is when it replicates inside of the host cell bursts. That's called lysis, and undergoes apoptosis or programmed cell death or cell suicide. And then all of the virions of the cold virus are released.